And we're back at the Future Directions in Choline with my co-host, Tom Druk. Our guest for this session is Dr. Bryan White. He's Professor Emeritus from the University of Illinois. Now, this week we've been having a 2-day symposium. We're in the middle of that. After the symposium, we're going to have a scientific advisory board, and Bryan's going to be giving a talk to that advisory board. Bryan, why don't you give us just a kind of brief overview, kind of the big rocks of what your presentation is going to entail?
Sure. So the microbiome has been implied into TMAO production, and then the negative effects of TMAO that primarily in cardiovascular disease. That cardiovascular disease side of the story is way outside of my expertise. My expertise is in the microbiome and the gut. And so the question comes, what is the role of the gut in the production of TMAO that gets into the bloodstream of humans? And so there's been a lot of work out of primarily Stan Hazen's lab that's shown a few things that are indisputable. So the indisputable thing is that microbes produce TMA.
Yeah.
TMA is taken up across the gut wall, and then in the liver is converted to TMAO.
Okay.
Can't dispute that. What you can dispute from the literature is that there's a diet effect on the production of TMAO, and that effect of that diet changes the microbiome in such a way that more TMAO is produced in the bloodstream. Okay. And when I've looked at the literature for this effect, there's a couple of published papers that really were not reviewed very well.
Okay.
So the manuscript in most questions is the Coates 2013 paper, and Figure 2 is really very poorly reviewed, and the data in that I have taken from their small read archive. So I look at the microbiome. The microbiome is a community, and you can ask a couple of questions. You can ask 4 questions really. You can ask who's there. You can ask how many of them are there. You can ask what can they do. That's genetic potential. Mm-hmm. And then you can ask what do they do. Mm-hmm. And the simplest thing to do is to find out who's there and, in a rough sense, how many are there. That's using 16S ribosomal RNA technology, which is what is done in the Coates paper. And so they took individuals given different diets, omnivores versus herbivores or versus vegetarians, and asked who is there and how many are there. And then they tried to correlate that to blood levels of TMAO. And in the paper, it looks pretty reasonably clear that there's a correlation between diet and the blood levels. But if you take apart that Figure 2 carefully, and then I took their small read archive, the sequencing that they did of 16S ribosomal RNA, and I did the analysis myself, and I got different results. And my results would be completely opposite of theirs, that there's a diet effect and that there's a correlation between the microbiome in those diets and blood levels of TMAO. So through that exercise, I've contacted the authors. The authors have, in some cases, been cooperative in saying, yes, we can give you data, which is fine. Don't necessarily agree with my results. They said I did different methodologies. So I went back and I did their methodology. Now the problem is that paper was done in 2013. I can do probably 80% of exactly what they did. The other 20%, because databases have changed, technologies have changed, I can't reproduce. But the bulk data does not match their data. There's no correlation between diet, and there's no correlation between TMAO levels in the blood with their cohorts.
Would you say, Dr. White, that it's is it you now have finer tools to analyze, or was it a matter of with what they had at the time, wasn't necessarily drawing the same conclusions, or have you learned things since, I guess is my question.
We have learned things since, but they, to me, and I've been doing this since 1987, before a lot of people were doing it, they didn't present two critical pieces of data, or one critical piece of data, let's say. And these are called PCOAs, and they are dots. The dots in space, if you've looked at a microbiome paper, they're the dots in space, and you draw a circle around the dots in space, and you can get two distinct sets of dots in space. Those PCOAs are based upon sequence similarity between communities. They did not present those PCOAs. They did a rough statistical model, which you can do on a computer, and it throws out numbers and says they're different. Okay. I never publish a paper, submit a paper, and this goes back to before they submitted theirs, without PCOAs. If your PCOAs don't show clear circles, then there's no difference. And when I redid their data and then generated PCOAs, there's no difference. And the statistics show there's no difference. It's a finer level of analysis, but they had all the data to do it. It's literally a push of a button and about 48 hours of CPU time, unsupervised. Why what they say they did not do that, they just used the statistical model. Why they didn't do the PCOAs, I have no idea. But if you saw those, you'd go, nope, sorry.
So it's created a bit of a controversy.
A big controversy.
Yes, and so what are the next steps to get to the bottom of this, right? Because I'm assuming they're saying they're right, and you're saying you're not so sure. And
so the literature since that is mixed as well.
okay,
I mean, you can find probably as many papers out in the literature that say that TMAO levels in the serum is linked to the biome as you can say is not. Again, the indisputable thing is that microbes in the gut produce TMA.
Yeah.
But can you correlate that to blood levels? And I haven't seen much in the literature that convinces me that the community structure, the presence of those organisms that do produce TMA is correlated or associated with blood levels of TMAO. Mm-hmm. So it's the Hazen group has taken their set of data and they've gone to march with it. And there's plenty of data out in the literature which is contradictory to that, but they're going with their story.
Yeah.
Now, he just recently published a paper, and I'll present a little bit of this at the SAB meeting, where he tried to correlate or do predictive modeling of the organisms in the gut and their genes, and try to say, can we use a study where we have cohorts that have high TMAO versus low TMAO, and can we look at the gene content and the organism content in the gut, this community structure analysis, who's there and what's their potential to predict blood levels of TMAO. And guess what? They couldn't. Now, the fallback is you're not looking at activity, which is the last piece of that puzzle, just because you don't have a correlation of the gene content,
Right.
doesn't mean you don't have a correlation of the activity. I would suspect that they're not going to find that if they look at it.
Yeah.
Well, this has been super fascinating. I think we're going to hear a lot of other talks about TMAO and some of the causality versus the association and all that. But it's fantastic to hear some of this deep dive into the microbiome piece, which clearly is your area of expertise. So
Well,
we appreciate
one thing with almost all the microbiome studies out there, causation is the gorilla in the room. And there's really only one disease that has been absolutely correlated to the gut microbiome, and that's C. diff.
Okay.
All the rest of them, there's associations, but the causation route has never been defined for any other disease. So is it the microbiome that causes the disease, or does the disease change the microbiome? And the only one that's been established for is C. diff, out of all the ones that are out there. So
It's great
Interesting.
context.
Yeah. We thank you for your time today. Thank you for being a part of our advisory board, and look forward to talking to you later on today.
Thanks very much.
Thank you.
Appreciate it. Thank you.